Thank you very much, Dan. I don't think I need to remind you that today is the first round in the World Championship in chess between a Norwegian Magnus Carlsen and a Russian Sergei Karyakin. It held here in New York. And I like to compare chess and science. In chess, you are contemplating about the position to try to find the right move to try to conquer the enemy king. And in science, we are doing the same thing to try to conquer the secret of life. So I'm coming from the northernmost university of the world. It's located on a small island, and it's surrounded by a very harsh landscape full of many peaks. It's a very desolate place, and we need a lot of international collaboration. And when people come to visit us, we are taking them to some of the peaks. Here is Brian Winchester from the Children's Hospital in London, and Pirkko Heikenheimer from the University of Helsinki. And they came in connection with a study in Tromsø on a lysosomal protein deficiency called alpha-mannosidosis, which I encountered in 1993 when I met a medical doctor at the university with 2 children affected with this disease, and I started to study it. 8 years later, I became coordinator of the EU consortium, the first EU project on a lysosomal protein deficiency. And you see here that the importance of this collaboration was that we included scientists working both in the basic science, also in clinical science, and where we also had an industry partner, Simonex, who produced an enzyme drug, which is not yet, but soon is coming on the market, called Lamocim. Alpha-mannosidosis is a lysosomal protein deficiency, and we tried to, during this talk, to convince you that ADA2 is also a lysosomal protein deficiency. As you have seen in the previous talks, ADA2, adenosine deaminase 2, is thought to be secreted from macrophages and being a growth factor, or it's binding to heparan sulfate proteoglycans on endothelial cells, and they're converting adenosine into inosine and affecting the intracellular signaling through the adenosine receptor. I have purified adenosine deaminase 2 from porcine brain, and it's a 58 kilodalton protein. You can sequence it by mass spectrometry. And in blue, you see the N-glycosylation sites. It contains 3 N-glycosylation sites. It means that N-glycosylated, and all secretory proteins are, almost all are N-glycosylated, so it's quite normal for a secretory protein. We can analyze the glycan structures by mass spectrometry. I will not go into details here. It's quite a cumbersome method. And we have studied from the porcine brain. We have studied the glycan structures from about 10 lysosomal proteins in the upper side here in red, and 13 extracellular proteins. I don't know, yeah. Well, you can see that on the right side there, you can see the blue columns, which symbolizes the extracellular proteins, and the red columns, the lysosomal proteins. And you see the lysosomal glycans are smaller. And the structure is actually a core-fucosylated paucimonocytic structure, and that's only found in insects. It cannot be produced in any vertebrates. So what does happen is that proteins that get into the lysosomes are trimmed by lysosomal exoglycosidases, producing unique structures in a lysosome. If you look at ADA2, you can see at one glycosylation site, which is circled down on the right side, you see the same insect-like structures. It shows that the protein has been transported to the lysosomes. It's a lysosomal protein. I don't know if I have convinced you, but I also have studied the charge and glycan structures located on ADA2. And on secretory proteins, the charged glycans are usually sialylated, and in lysosomal hydrolases, they are usually containing mono-6-phosphate sorting signal. And if you look at ADA2, which is in green here, you see that all the structures, at all the glycosylation sites, there are mono-6-phosphorylated glycans. And you see also that the distribution of the mono-6-phosphorylated glycans are exactly the same as the lysosomal protein, which is in red, meaning that the protein is sorted to the lysosomes in a transcontinental network, and it is not secreted. And you can ask yourself, but the protein is secreted from the macrophages. If you look at the literature, you will find papers showing that the mono-6-phosphorylated glycoproteins are secreted in activated macrophages. So it's no discrepancy between these results and the fact that the protein is secreted in the macrophages. So you can think about what is the role of this secretion. And I think that maybe one of the roles is to transport the lysosomal hydrolases to the early phagosomes, so we can get an early hydrolysis of particles that need to be phagocytosed. We have examples of diseases of lysosomal proteins, which are causing autoinflammatory diseases. Here in mice, lacking a protein called lysosomal DNase2 and causing a defect in a degradation of nucleotides in lysosomes. The macrophages from these mice are secreting cytokines like TNF-alpha and interleukin-1 beta, which is causing this rheumatoid arthritis. So can ADA2 have a function in lysosomes? And if you look at the structure of the binding of the enzyme with heparan sulfate, which is quite known, and Savialo was talking about it, you see that both DNA and RNA have the similar kind of structure. And actually, very many proteins which bind proteoglycans will also bind to helical DNA and RNA, indicating that the protein can be a DNA-binding protein. So as a conclusion here, we can say that we know that the defects in lysosomal nucleotide metabolism can cause the secretion of cytokines from macrophages and result in autoinflammation. And we know that ADA2 is a lysosomal hydrolase. It can transform adenosine to inosine at acid pH, which is a pH which occurs in lysosomes, and it may bind to DNA and RNA. So what is the next move? I think we have find many more new aspects, and I think the important next move, I think, is a collaboration between all the disciplines, which you are, both in the basic science up to the clinical science, and finally to try to find a drug that can kind of target the primary causes of DADA2. Thank you very much.